Gary, thanks for lining this up and getting
This meeting is being recorded.
Thanks, Gary, very much, and for lining up this talk and an audience to hear my tale of two fishes. I'll be talking about some recent work that we've done with Atlantic halibut and Atlantic wolffish. I've identified as the co-authors for this particular talk as Chris McGuire with the Nature Conservancy and Elizabeth Fairchild with the University of New Hampshire. They were the main PIs that wrote some NOAA grants to get this work started, really off the ground. And but there's many people I want to acknowledge for helping. I'm going to put up just about everyone here. Obviously, we were working with the commercial fishermen. So we had halibut fishing partners, particularly the Cape Cod Commercial Fishing Alliance. George Maynard and Rachel Marshall were associated with them at the time of these studies. And the Wolffish fishing partner was Captain Jim Ford, and he's in the Gulf of Maine. And we reached out, and the idea of enhanced fishing, biological sampling is to connect with anyone that really can supply samples at the times of the year and the locations that we're looking for. So we reached out through your agency, the Massachusetts Division of Marine Fisheries, through their surveys, also their commercial fishermen in Maine, our Northeast Fisheries Science Center surveys, the bottom ground fish survey, the longline survey, the study fleet that we have. And of course, those samples were in many cases processed in the field by fishermen, but there's plenty of sample processing by a number of individuals listed there, including Scott Elsey with the state of Massachusetts, who did the aging for both species, as we'll talk about in a minute. And finally, this funding, as I said, really got off the ground through two separate Saltonstall Kennedy grants. That's a NOAA funding program for both the halibut and the wolffish identified there. So a lot of people involved in this that I'm happy to share our results. From a personal perspective, everything I needed to know about this project, I learned it when I was a child from Aesop's Fable, or where I learned most of the Aesop's Fable were through Fractured Fairy Tales on the Bullwinkle cartoon hour. The story of the goose and the golden egg is a really good one that I think most people are familiar with, and it's really easy to follow. If you kill that goose to look for more eggs inside, then you've killed you stop the opportunity to get one on a steady basis. Of course, it's a little bit harder to apply with fish. The relationship of how many eggs fish produce is much higher. It's often very difficult to measure, but in many ways it boils down to simply let fish spawn at least once before they are harvested is a good kind of rule of thumb in terms of thinking about where to start the discussion of sustainable fishery management. But in many cases, we actually don't know the age or size at maturity for these fishes. And that's where we were starting with this. There was uncertainty about this information, basic information about the maturation schedule for these fishes. And so that's where, that was one of the driving forces that I had in mind as I got involved in it. So I'm going to present actually first a little bit of information about halibut. Then I'm going to present some information more about the wolffish study. And then I'm going to kind of show you how that we looked at the maturity estimation and how we applied that for both species. We'll start with Atlantic halibut sampling, which occurred in the Gulf of Maine. There was, I think you can see the little arrow. Let's see, find this arrow here off the elbow of Cape Cod. We see these dark circles that these are samples that were primarily from the Cape Cod Commercial Fishermen's Alliance. They were using a variety of gears, gill nets, long line. There was broad sampling throughout the Gulf of Maine. These open triangles are from different fishery independent surveys like the Northeast Fishery Bottom Trawl Survey. I think the Massachusetts IBS, the industry-based survey, collected a number of fish. Here in the central portions of the Gulf of Maine, the Northeast Fishery Science Center's longline survey. So we had a lot of different sources contributing samples from throughout the Gulf of Maine. This graph really shows you the influence of the SK grant that was awarded in 2016. Up to that point, we were relying just on the bottom trawl survey just to kind of get a baseline of samples shown there in yellow starting in 2014. We were getting less than a couple dozen fish. They were decent size range, but that really wasn't enough to get going. But the SK funding really increased the sampling in 2017 as well as 2018, ultimately more than doubling our overall sample size. Not just in the overall sample size, but really covering also, you can see these dark purple bars in the month plot on the right, where we were able to increase sampling coverage very nicely, except for say March and very low samples in December. But this kind of broad monthly coverage was very important, as you'll see later. I'm not going to talk too much about the age and growth information, but I want to show its contribution. Atlantic halibut, as most flatfishes, did exhibit dimorphic growth. Scott and his team spent the time looking at the effect of whole ages from whole versus section otoliths, and were able to show, not surprisingly, that section otoliths were worth the trouble and provided more reliable ages. And not surprisingly, there was the sex-specific von Bertalanffy growth equation was the best model fit for the Atlantic halibut, with females growing to a larger size at age across, particularly at older ages. What's interesting here, the context of the fishery, is that it seems to be actually increasing, but it's still at very low levels overall. It's not quite what's happening, the explosive, sustainable, certified fishery in Canada, where the fish can easily get double the ages as they are in the US waters. But now we've got a very nice documentation of where we are in the 2010s from this study. I'm going to focus a little bit more on reproduction, which is more my specialty. And I'm going to look at how to show how the reproductive seasonality was evident from the samples. I'm going to start by just plotting the GSI, which is the gonadosomatic index, which is basically the gonad weight ratioed to the ovary-free body weight in this case. The idea is, as the fish is growing, its gonad is growing, but you want a ratio to the body weight to pull out the effect of just the fish growing. You can see in this plot here with this blue line, the individual GSIs from individual fish is increasing here. And this is the essence of enhanced biological sampling. If we just relied on our spring and fall surveys, we would only capture a part of that increase. And we're able to see that a little bit better by having some samples in the middle. These low numbers are from immature, resting, or skipping females, depending on the season or the individual condition. But you get a sense that that in the US waters, it looks like the spawning is occurring in the winter. It's estimated from a Canadian study to occur in November and December. And so you can get a sense that the gonad is increasing in weight as the gonad is provisioning yolk to the oocytes and ultimately getting ready to spawn. We examine the gonads directly, macroscopically. So these images show what's clearly, these are one centimeter cubed reference lines. You can see what's very clear, an immature fish at 70 centimeters total length, and what's easily observable as a mature fish with highly vascularized gonad wall, the manila color to the surface. You can see if you take a close-up look, the oocyte sizes are starting to get a big. The individual germ cells are getting to be larger than 1 millimeter in diameter, but the ones in the middle are difficult to understand a little bit. They seem to be getting bigger, but it's not always clear what the macroscopic stage is. But we had some gonad, we looked at gonad histology too, which really helped us identify, sort out, yes, these are immature ovaries. If you look at the histology, this is a thin section that's been stained from the gonad, and you can see these individual cells with their individual nuclei. These are primary growth oocytes. There's nothing in the cytoplasm indicating any hormonal activity. If you go to the other extreme, the mature oocytes, these are still those primary growth oocytes here in the light purple, but you can see these much larger cells here with the nucleus here, and in the cytoplasm, they're actually showing evidence of yolk provisioning. So the female is provisioning its young cells, the haploid cells, with yolk that will be used as the yolk sac for nutrition in the early yolk sac larvae. And in the middle here, we would score these for the purpose of maturity estimation as immature, but they're showing a physiological maturing going on. Namely, here's the oocyte here, and this purple ring around the distal edge of the cytoplasm are the cortical alveoli, which is actually something that's to prevent more than one sperm fertilizing an egg later on. So it's indicating some hormonal activity, but no provisioning of the yolk to indicate that the fish is going to spawn in the coming season. We also noticed skip spawning. This is an individual that was identified as a skip spawner. It was collected in October. It had a low GSI, but it was higher than the immature fish, had a very thick gonad wall. It was primarily the fact that it was collected in October and it was just starting vitellogenesis, but the provisioning of yolk, and that occurs over the process of a whole year. So this is a fish that actually is not spawning in the next season, in that winter season. And one of the things that we would do to identify the differences here on the right, we would see, for example, comparing an immature to a mature fish, is the gonad wall here is very thin. All these little connective tissue that go into the lamellae of the germ cells is thin, indicating that it is not spawned and it's not ready to spawn anytime soon. Here, this is the fish is physiologically mature, but it's not ready to spawn yet, but it has this thick gonad wall indicating that it's spawned in the past. And that's really the definition of skip spawning. It's spawned once in the past, but it's not skipping, it's skipping 1 year in particular that you're measuring. And this is most easily observed, most commonly observed in young fish that have sort of energetically started to spawn, but they can't do it every year, at least until they get bigger and more energetically enriched. So this leads us to a proposed reproductive life cycle for Atlantic halibut. We're using basically the scheme that's used at the Northeast Fisheries Science Center, where you're immature once, and once you become mature, you stay mature, although you may skip. So there's this functional maturity here, and skipping, which is a disassociation for some fish between the mature and the spawning biomass. The preliminary estimates from our study indicated two different ways that we could look at it. From 4% to 25% of the mature females were skipping in our sample. So that's just a quick peek at the sort of the outline of the reproductive biology and the biology of Atlantic halibut. I'm going to also do the same for Atlantic wolffish, which we also sampled in the Gulf of Maine. You can see that we had sampling throughout the shelf, or throughout the Gulf of Maine, the South Channel, Northeast Peak, well into the Bay, well into the Gulf of Maine, in deep areas here in the open. These dark circles in this case indicate the long line. The shaded area is the study fleet area. There was concentrated sampling by some permitted fishing by the University of New Hampshire and Captain Jim Ford off of Cape Ann in the Stellwagen Bank region. And they produced a tremendous amount of helpful samples for our coverage. Again, this award went out from the Saltonstall Kennedy program in the same cycle in 2016. Our sampling was specifically in 2017 by the University of New Hampshire and Jim Ford's cooperative trawl, which again increased our samples tremendously. And not only just another contribution to the total number, but really what they were able to do was capture this summer season, which was very helpful in terms of connecting where the oocytes and the reproductive cycle was in the spring versus the fall. We were able to cover this summer season because of the New Hampshire trawl. Scott's group contributed the Atlantic wolffish aging, showing again dimorphic growth. In this case, the males reached a larger size, and as Gary found for his study on wolffish aging growth, the Gompertz model beat out the, if you can say that in a statistical sense, the von Bertalanffy growth equation shown here for the males to be larger at sizes than the females. Again, he used sectioned otoliths. In this case, they had done a previous study that was published in Fishery Bulletin 2015, I think. Fairchild et al. Scott had already worked out that the section otoliths were better than the whole otoliths. Very nice contribution to have the ages. Again, I'm going to focus on the reproduction, which is my specialty. And we actually had very good sample sizes for these months, not the winter, but the months from April to November, showing that the ovaries increased in size. These are the mature fish only, showing this strong trend. You can see a peak in the ratioed gonad weight to the body weight, and then a drop in October, indicating that spawning was well underway and over by November. We found this very interesting. In the fall season, we could see a difference between fish that were physiological versus functionally mature. So for example, these are photographs that were taken on our groundfish survey here, in this case, for a fish that was under the size of median maturity versus a fish that was over the size of median maturity. This is a physiologically mature fish, but has a very low GSI and very small oocyte diameter of 1 millimeter. Of course, some of you might be thinking 1 millimeter is actually quite a large fish egg. And that's true for most marine fish, but in fact, actually for wolffish, the oocyte, the ripe egg is actually 4 millimeters in diameter. You can see here for a functionally mature fish in the fall, you should see a gonad ratioed weight that represents something like 17% of the body mass, and the oocyte diameters, as you can see here. You can see these 1 centimeter hatch marks right here. You can see these oocytes, even before they've actually been released into the lumen, are really quite large. What we looked at, we thought this was a very curious fish, that this fish was, the gonad was pretty well along the way, but it was certainly different than this. This begged the question of what was going on. What we observed there was, in the spring, we didn't observe that. We only observed that small ripe gonad in the fall. What was happening in the spring, here's a picture of the gonad. These are individual germ cells that actually are in vitellogenesis. That means the yolk these are yolk globules in the cytoplasm surrounding the nucleus. And we can see here by the fall, for some fish, those cells that were vitellogenic have gone bad, and they're marked with an asterisk, red asterisk here, indicating massive atresia, massive cell death, whereas these cells are actually for the next year spawning. Massive atresia was not just evident in the fall, it was most likely in the smaller size fish. So in the spring, you saw a number of fish that were mature across size range from as small as under 30 centimeters, but most likely between 50 and 70, and then some is larger than 1 meter long. But in the fall, you start to see these individuals with the small gonads that have massive atresia, and they are statistically smaller than the other fish that are actually going full, that are going to complete maturation and spawning in the spring. So our proposed reproductive life cycle for Atlantic wolffish is a little bit more complicated than that for halibut. We see a physiological maturity that can be observed in the spring that may lead to functional maturity or may lead to this abortive maturation. Presumably that might start, that in other words, not a skipping so much, but a delay of maturation after an initial failed attempt and resorption of the yolk cohort. Functional maturity leads, once mature, you stay in this cycle unless you skip. And we did notice one female in our collections that was skipping that amounted to less than 5% mature skipping for the mature females. But we did also notice that. So there I gave you kind of a more sense of the dynamics, sort of what we looked at, how we interpreted it, and how we concluded the mature life cycle was. Going forward, we often just take that maturity data and put it in a binary context. The fish is either immature, as in above the red line, or it's mature below that red line for both these fishes. We're going to actually walk you through how I would do an ogive estimation. First, you take the total number of fish and you plot the data. So in the case for Atlantic halibut, we had 313 females. And those that were mature, we're plotting here. These tick marks are indicating their fish length in centimeters. And these tick marks here are indicating the immature fish. We take a logistic model, which has a S-shape, sigmoidal shape that can fit to the data using this function here. There is a linear component to this model. So it makes the interpretation pretty easy. For example, we can look at the 50% maturity here and extrapolate that to get the L50, the length, the median length at maturity for Atlantic halibut was 130 centimeters or 51 inches. We can also put, in this case with bootstrapping statistics, we can put a 95% confidence limits around it. So we feel pretty confident that the L50 is somewhere between 125 and 135 centimeters. Using this data, having a 130 centimeter reference point, we can, since this halibut is a discard fishery, one proposal that we're using in our manuscript is to propose that you can monitor the proportion of mature females that are caught each year in this bycatch fishery, which Dan Hennon looked into closely from the commercial data to show that this proportion is actually increasing, where it was under 20% in 2009, and 11 years later, it's over 40%. Now, there's a lot of assumptions to interpreting this, but this is something that can go into a data poor, a stock assessment as one indication that the fish are getting larger overall under the current environmental and fishing conditions. We can also do that maturity estimation for Atlantic halibut, median age at maturity, which turns out to be 9.5 years, in the confidence limit somewhere between 9 and 10.7 years. So Atlantic halibut, one of the challenges of letting letting the fish grow to spawn once is that it's at an individual level that's taking approximately 10 years. How may this information be used for wolffish? A little bit slightly more complicated, but if you recall back to 2007 was an early attempt to look at the maturity in an early assessment, they were using the macroscopic evaluations of maturity, and some of those were all over the board. And a lot of it was the inability to recognize this abortive maturation that from the macroscopic observations. The histology has helped. I think actually we can do better with macroscopic evaluations, and we're continuing to do the gonad histology just to make sure. But they were so uncertain back 15 years ago that they were using as a reference point 3 different L50s, from 40 to 75 centimeters, because they didn't know entirely which one was correct. In Canada, where it's colder, not surprisingly, wolffish get to be larger. Their L50 is larger than we're estimating for the US waters, but we didn't know what that was here. But using gonad histology leads to a more accurate and precise estimate, which we're publishing as approximately 53 centimeters or 21 inches. Somewhere with 95% confidence limits between 49 and 55 centimeters. And this was used as the reference point in the 2020 stock assessment. We also know it takes years for wolffish to mature, like the Atlantic halibut, not nearly as long, but the A50 was estimated at 6.7 years, somewhere between 6.2 and 7.2, which is not bad for a fish that lives its entire life less than 50 degrees Fahrenheit. So in conclusion, not surprisingly, well, maybe it might be surprising to some people. I mean, the US Northeast, one of the most data-rich ecosystems in the world, there are many data-poor species whose management would benefit for improvements to basic life history. And I hope I've demonstrated that the case for these 2 fish. But also that we did improve the life history information. We certainly were benefited by working with fishermen who have skills to get more data, and in this case, more samples into the stock assessments. It presents challenges requiring meetings, exempted fishing permits, and oversight, but this engagement also led to shared understanding and even new insights. We worked closely with the modelers, Dan Hennon for halibut and Chuck Adams for wolffish, to deliver the data in a form that they could readily uptake in recent and future stock assessments. And that is what I have for this talk today. Thank you.
Thanks, Rich. That was really interesting. Are there questions for Rich? You can either unmute yourself and ask, or you can just throw it in the chat. And there is a question in the chat from Bob. Sorry if I butchered your last name, Bori. Can you tell us what percentage of mature fish, either species, are in the skipping phase at any one time? Any thoughts as to the cause of skipping?
Yeah. So far, our estimates for wolffish was quite low, less than 5%. And there were 2 ways to estimate it for halibut, and one was as low, was less than 5% or about 5%. The other one was a little bit higher, 25%. I would say all of these I would consider preliminary, because they're really the first time anyone's ever done that. There are some very nutrient poor lake systems, freshwater lake systems, where fishes can be shown to have skipping 20, 30%. Winter flounder off Labrador, which is really at the extreme of its northern distribution, can have rates 20, 25% skipping. And these would be significant skipping rates if they're true. I don't think the Gulf of Maine I'm not expecting the Gulf of Maine has that high, but certainly we've demonstrated that it's not 0. I would think that my guess is it's probably something less than 10%, but it would be nice to sort of get more samples now that we're more proficient, and continue this in the future at some level. Also, to look at independent measures. I've talked with I know William DeVoe and others at the state of Maine are tagging a lot of fish. Atlantic halibut in particular, and there's some potential to get information on skipping rates from electronic tags that are going out. I know they've done that for Pacific halibut, a different species, but the rate can be something like 10% out there. I can't quite remember. So that there are some independent ways to estimate that, and that's something that we should be monitoring because the size at maturity, the age at maturity, the skipping rates are not necessarily fixed once you measure them, and they could change to, they could respond to climate or fishing conditions. So it's something to keep an eye on. Thanks for that question.
Micah.
Thanks, Rich. That was a great talk. Two really cool fish that I don't get to think about a lot. So I learned a lot. But I'm just curious about halibut. That I've been told that there's not a lot of evidence of spawning grounds in U.S. waters, and that they're sort of further afield. I'm just wondering how that might influence sort of the estimated maturity ogive if the ones that have recruited to the spawning stock, if that's outside of our sampling, whether we're not sort of biasing that maturity ogive towards late bloomers.
Yeah, I mean, there is a concern about that. I mean, that's not restricted to these two species. I think the two species represent different examples, whereas the spawning grounds for wolffish are more local in the Gulf of Maine, whereas what we do know about Atlantic halibut is that they go into deep waters on the shelf slope to spawn, and obviously out of state waters, but in some cases to the extreme of the federal waters. So it's important that we sample at different times of the year to get some represented. I mean, obviously, if you don't have any, if you're relying on sampling on the spawning grounds for Atlantic halibut, we're not, as far as I know, we're not sampling on the spawning grounds, but we can sample before the fish migrate to spawn, and then you're relying on an unknown about how well mixed they are where you're sampling. And as a data poor species, we have to imagine that we don't really know what sampling design is perfect. So we sort of go with that as a start. So I think one of the best ways to do it right now is to cast a white net with lots of gears, different geographic locations, different seasons, and try to mash it all together. But given more control over the sampling design with better life history information, that'd be great.
Thanks, Rich.
You're welcome.
Sandy Sutherland asked a question in chat. Can you explain why wolffish had an A50 of 7 years, but growth didn't slow until about 15 years?
So, oh yeah, that's a good point. Generally, I don't know. I haven't taken the differential to see where growth is slowing exactly, but you often associate sexual maturity with a slowing of the growth. There's many things about wolffish that are curious in terms of their internal fertilization, with the males being larger and having nesting behavior, them losing their teeth at one time of year, all en masse. So there's probably a few fingers to point there, but I don't know I don't have an easy answer for that, Sandy. Thanks for bringing that up.
Anna Dorrance asked, is there a size after which you no longer observe skipping or see it at a much lower rate in halibut, or specific years you saw less skipping?
Yeah, Our goal, I'm very happy with our goal with observing skipping, but those are the next kinds of questions that would be useful to answer. I'm not sure we're going to have the samples to really look at that, but based on other species where they've studied skipping more intensely, it's really a matter of the bioenergetics. And so for what you generally see the skipping after, the wolffish, you see an issue of delayed maturity through this abortive maturation. But once you get through that and you do actually mature, the skipping is generally in those early years where the fish had enough energy to spawn 1 year, but not necessarily its first year, but not necessarily the second and the third year necessarily, because it exhausted itself. So you generally see it for a few years, and then they reach this period where they presumably don't skip very much unless there's some real nutrient condition that happens periodically. But the problem with many fishes in working in this environment is their size and age truncation to a point that it's really hard to generalize that for marine fishes that we exploit in the North Atlantic, because generally they just don't live long enough to really completely test that hypothesis.
Let's see, any other questions, Rich? Can you unmute yourself? Hearing none, I just want to thank Rich for taking time to give us a great presentation. We appreciate your time. And sorry, Chris McGuire has a question. He just raised his hand.
Hi, Chris.
Hi, I don't actually have a question. I just wanted to take this opportunity to thank Scott Elzy and the Aging Growth Lab. Rich mentioned this, but just as the project PI, Scott and the Age Lab's contributions to the Halibut project were tremendously important. And so just a big thank you to Mass CMF and Scott and his team for the work in contributing to what Rich presented here today. So thanks a lot for that.
Right. All right. So thanks again, Rich. So the next talk is 2 weeks from today. I think it's February 18th. 15th. Yeah, let me pull up my schedule here. Hold on a sec. Sorry. 15th. Yep. February 15th. And Story Reed from our division will be talking about. Well, his title of his talk is Port by Port: Profiles and Analysis of the Massachusetts Commercial Fisheries. So come on back, and we appreciate you attending. And if you want to be on our mailing list, our email mailing list, just send me an email at gary.nelson@mass.gov, and I'll put you on the mailing list. All right.
Thanks for this opportunity, Gary.
All right. Thanks, Rich. Appreciate it. Bye, everyone. Have a good day.